NIH, at a time of hiring freeze, which was done on the first day by our current President Trump. I couldn't figure out which one was Trump. So let's see where the up and down is. That's weird. I guess I'll use this. So this is a recent award that Peter received from Doug. Presenting it to him. This is sort of the end of his career receiving an award, and a well-deserved of many that you'll hear about in the next 10 minutes or so. And here's sort of closer to the beginning of Peter's career. This was a few years after Peter came on board. In 81 you came on board, right? We hired you in 80 and you came on 81? 1981, yeah. About a year after we committed to him. And I'll tell you the story, and I'm sure he'll tell you the story at the end. So this is Peter and Daphne at a crab feast in my backyard, which we used to have to welcome people to Maryland. And also shown are Sue Jakin, who was Peter's postdoc, and Jim Stevens, who was a postdoc with Luigi DeLuca. And they met here in the lab and ended up being married. And one of many examples of lab romance. Not too many examples. An example. So, how do you summarize a career like Peter's, which has been so dramatic in many ways? Peter, I think, uniquely among most of us successful scientists, and really made conceptual discoveries in two fields of research that really changed the complete direction of that research. I think that's extremely unusual. And he did it in a way that I think is very typical of NIH and maybe unique to NIH. And that'll come through, I think, as we go along with this presentation. So there's several ways to summarize a career. One is like this with numbers, and I'll go through those numbers, and the other is sort of historical, showing you the kinds of thinking and the kinds of directions that Peter went through during his three plus decades at NIH. So the breakthroughs that can be attributed directly to Peter's work are the breakthroughs in PKC and in vanilloid pharmacology. And they've led to new therapies for cancer and pain and other diseases. We will never, ever discount Peter's productivity because it carried us through 3 site visits. While the rest of us published 2 or 3 papers a year, Peter was publishing 10 or 12. When we presented the work of LCBG, we always just added all the papers together, and the Sally, don't tell anybody about it. And it looked like a huge number of papers thanks to Peter. But in the end, there were 475 publications, 21 patents, and more than $1 million in licensing fees were brought into the NCI. Mentoring 49 postdoctoral alumni, many of whom have very successful careers throughout the world. Some of whom are here today, happy to say, and hopefully they'll say a few words about Peter. Many students and inspiring opportunities for young scientists. Peter has won many awards, too many to put all on a slide, and we'll talk about several of them individually as we go along. And of course, from his many contributions to research, he received considerable national and local recognition, being on boards and workshops and so forth. So that's the numbers. But what about the history? So let's start going through the history. So the history starts with the most important model in cancer research, that of mouse skin. So in the late 70s, there were basically 2 major areas of carcinogenesis research. One was viral oncogenesis, which there was a tremendous amount of activity, most of it related to very potent viral oncogenes, and one step transformation, which really didn't fit in with cancer development in humans. But of course ended up revealing oncogenes and have been extremely important in cancer research since then. The other was multi-stage models, mostly chemical carcinogenesis models, and the one that really pioneered that area was the induction of tumors on mouse skin, where the terms initiation, promotion, progression, lytic conversion all came from. So the terminology of multi-stage carcinogenesis came from that one. Nobody knew anything about oncogenes and chemical carcinogenesis at that point. We knew chemicals reacted with DNA. But there was a lot of attention to tumor promotion, which was a reversible process. Today we call it microenvironment, but in those days we called it promotion. And the most potent promoter were the four-ball esters. Peter was a associate professor at Harvard at that time and began to study the four-ball esters, mostly in cell culture models. And I was studying it in skin. And there were a lot of meetings. When a topic is hot, there are always a lot of meetings. And so Peter and I met at these meetings and were very interested in each other's work. We started talking about Peter coming to the NIH and joining the lab that I was the chief of at that time. And we finally sealed the deal, and then Reagan came in, and that's the end of that. For about a year, Peter's postdocs came first because we could hire non-government employees. And then Peter finally got here. And it's just been a joy working with him ever since. We've collaborated on over many years on many topics, and it's just been a terrific thing. So going through 475 publications and trying to pick out the top ones is ridiculous. I can't do that. So I decided to go by decade and pick out just a few examples. Maybe not even the most important examples, but the ones that my brain would allow me to remember. And show you the progression of this career of his and this research program of his over the years. So the 1980s, when Peter joined NIH, was the year of the discovery of the 4-ball ester receptor. I cannot tell you, I can't emphasize enough how important this discovery was. Not just for tumor promotion in the skin, but for cell biology in general. Because people have been studying 4-ball esters in all kinds of models, and they did all kinds of things. It was very, very difficult to put together a story of how 4-ball esters led to tumor formation. It was really almost impossible. But Peter and Paul Dredger, one of his postdocs at the time, and then others afterwards, showed for the very first time that there was a receptor for the 4-ball esters. Mostly working in cell culture models and then going into skin and other models. He showed very specific binding using a very clever tool of tritium-labeled 4-ball dibutyrate. And these two papers, you can understand how a dramatic change takes place in understanding from one or two experiments. These papers revealed the biological basis for the entire protein kinase C family. So PKC was an unknown enzyme at the time, not studied well. And the discovery of protein kinase C as the receptor for the 4-ball esters revealed a whole panoply of cell biology that previously was not understood. It was a tremendous, tremendous breakthrough in the field. He went on during the 80s to continue searching for endogenous PKC ligands, discovered that diacylglycerol was the endogenous ligand, and then studied other ligands for the 4-ball esters and really opened up the entire field. The first author on publication number 60 is Nancy Sharkey, who most of you know as Nancy Lewin. And Nancy is here. There she is, right here. So this was a huge breakthrough during this decade of the 80s. Peter was recognized nationally at that time by receiving the Rhodes Award from the American Association for Cancer Research. This is an award given to someone under the age of 41. I'll never get that award. And, And his picture was on the cover of Cancer Research at that time. And ironically, one of the other people on that cover, oops, I think I was going to do that. One of the other people on that cover was Bernie Weinstein, here. And Bernie was also working on tumor promotion in the colon. Not with Forbel esters, although he was working with Forbel esters as well. But he got the Clues Award that year. So it was a pretty hot topic. When we got into the 90s, he continued along this trail of understanding PKCs, understanding the structure-activity relationships. Became very important to him with Marcello. He was looking for specificity, and some of these other papers were trying to understand the binding domains of PKC, and trying to understand how the various, at this point, multiple PKCs were already, had already been evaluated, and trying to understand how this multi-isoform family could have so many diverse effects in cells and be differentially expressed and so forth. Continuing to be extremely important area of research. In the 90s also, he started working with Victor Marquez and in designing derivatives of diacylglycerol that might have specificity for specific isoforms of PKC, trying to, in a sense, in a pharmacologic way, which is Peter's specialty, to design ligands that might have targeted specificity for one isoform over another. Then things took an unusual